呢條題目你又覺得難定簡單呢？喺留言區留低你嘅睇法。喂，咁多位大家好，我係梁 Sir。解完 MC 之後呢，我哋又嚟解下長題目咯。2025年卷1月報第一條係有關於細胞分化嘅。當年嘅考生表現呢，考評局就話 good 喎，好喎，咁我咪一齊嚟睇下大家表現有幾好啦。呢條題目會唔會係其實容易答嘅呢？咁啊，題目啦就俾咗兩幅圖我哋啦，電子顯微鏡嘅圖像嚟嘅，就顯示咗一個 B 淋巴細胞喺佢分化之前同埋分化之後嘅外觀，cell differentiation 細胞分化。咁望到個樣啦，左手邊 B 淋巴細胞，右手邊呢個其實係咩細胞呢？就係我哋嘅漿細胞 plasma cell 嚟嘅。咁啊，啲細胞嘅標籤都俾咗你噶啦，細胞器啊，呢個係細胞核，呢個都係細胞核。咁其餘呢啲節啊、細細粒啊，呢啲咩結構嚟嘅呢？陣間題目都會問嘅。入題目咯噃 ，Part A 呢個就問啦。究竟啊，左手邊呢個 B 淋巴細胞，佢係會受到啲咩嘅刺激 stimulate， 從而啦就引發咗呢個分化呢？其實呢條題目啦就考緊我哋有關於 B 淋巴細胞究竟佢係點樣做一個特異性嘅免疫反應？呢個特異性嘅免疫反應就係講緊 B 細胞係會受到病原體嘅抗原去活化、去激化佢、刺激咗佢，然後就會產生唔同嘅反應去消滅病原體。咁之後就會講埋咩製造抗體嗰啲嘢啦。但係今次唔使講咁多，淨係講究竟無啦啦點解佢會活化咗、激化咗？其實佢就係受到抗原啦、病原體啦、疫苗啦、細菌或者病毒感染咧，就會刺激咗呢個 B 淋巴細胞噶啦。咁所以啦，無論你答抗原又好，或者答後面其他嘅答案咧，都係可以接受嘅，因為病原體嘅表面係會有抗原，打入你身體嘅疫苗都可以有抗原，甚至乎啦，真正受到一個病菌或者病毒嘅感染咧，你身體都係面對到呢啲抗原嘅。所以 Part A 就是這樣啦。去到 Part B 咯，喺呢個分化嘅過程當中，除咗細胞核之外，題目就要你講兩個喺幅圖當中可以觀察到嘅結構改變，而呢兩個結構改變係對於呢個 B 淋巴細胞當佢分化咗之後嘅功能係十分之重要嘅。咁題目自不然就考翻我哋傳視技能啦。首先你要指出嗰兩個可以觀察到嘅結構改變，然後描述下究竟點樣變呢？係嘛，變還變，咁你都要講點樣變噶嘛，係咪？然後啦，就考翻我哋有關於 B 淋巴細胞嘅功能啦，因為題目都講啊嘛，呢兩個改變係對 B 淋巴細胞分化咗之後嘅功能好重要啊嘛。咁究竟有幾咁重要啊？呢個係漿細胞，頭先提咗大家噶啦。漿細胞係會將由蛋白質所組成嘅抗體釋放去血液當中，去對抗尚未進入身體細胞嘅病原體。簡單啲嚟講，即係製造抗體啦。咁啊，但係個抗體係由蛋白質所造成嘅，又唔好忘記。所以其實題目係考翻我哋有關於 B 淋巴細胞嘅適應性特徵。喺呢個細胞當中，我哋見到啲咩嘅細胞器，係佢嘅結構上係有改變嘅呢？答案就係講緊線粒體嘅數量增加啦。你見到啦，分化之前同分化之後呢啲咁嘅細細粒呢啲咁嘅圓波仔咧，係咪多咗好多啊？線粒體數量增加，你要指出係線粒體有改變，而個改變係數量增加。然後啦，去到粗糙內質網咯，係咪好多呢啲接啊？佢嘅數量都係增加，所以啦，指出同埋要描述佢個轉變。然後去到第二部分啦，引申落去嘅啫。要你解釋下呢兩個嘅結構，即係線粒體啦，同埋粗糙內質網。喂，佢哋有變化噶嘛？係咪？係啊，有變化噶。佢哋呢兩個結構如何互相合作、互相配合，從而就令到呢個分化咗嘅 B 淋巴細胞能夠發揮到佢嘅功用 ，bring about the function。所以呢個題目係考緊我哋兩樣嘢。第一，你要做到功能上嘅配對。究竟啦，線粒體佢嘅功能係乜嘢呢？粗糙內質網佢嘅功能又係乜嘢呢？頭先我哋講啦，喂， 將細胞製造抗體喎，梁 Sir，而抗體係由蛋白質所造成嘅喎。OK，咁我就一定要噏得出粗糙內質網係攞嚟製造抗體，因為抗體係由蛋白質所組成㗎嘛。所以啦，粗糙內質網製造抗體呢個係佢嘅功能嚟嘅。而線粒體佢嘅功能係咩啊？提供能量，釋放能量。但係啦，係咪咁樣講完就算數啊？唔係。配對完功能之後，記住將各自嘅結構功能拉上關係，所以你要學識點樣表達你嘅意思啦。線粒體為咗粗糙內質網製造抗體嘅時候，佢哋提供能量，又或者用英文嘅理解啦 ，mitochondria provides or release energy。嗱，佢功能配對啦，係咪？The production of antibody at the rough endoplasmic reticulum， 嗱，都係功能配對啦。不過啦，呢啲能量，喂，俾邊個用㗎？就係 for the production of antibody at the rough endoplasmic reticulum 啦。所以啦，如果喺第二部分，你將呢兩句句子分開去寫，線粒體提供能量，句號，粗糙內質網產生抗體，句號，兩句分開寫嘅話咧，係0分嘅，因為你冇拉上關係。呢個就等同於我平時提大家做比較題嘅時候，一句過 A 係咁嘅時候，逗號，但是係嘛 B 就係咁，句號。咁所以你要明白到你究竟有冇將唔同嘅概念拉上關係，有冇睇清楚題目呢？呢一個就考你功夫咯。就快啲睇翻直線抽擊答題法啦。跟住啦，又嚟到一點出發啦。今次嘅題目係由 B 淋巴細胞開始，考我哋兩樣嘢。第一，分化；第二，細胞器。喺分化嘅概念當中，今次考我哋嘅漿細胞就梗係製造抗體啦。而記憶細胞都好緊要噶嘛，就係去記認嘅抗原。呢個咧我哋都要去學識嘅。今次問漿細胞，下次問記憶細胞得唔得啊？梗係可以啦。有關於記憶細胞，不得不提嘅梗係免疫記憶力啦。然後啦，就去到細胞器咯。今次想考我哋嘅咧，其實就係適應性特徵，亦都考我哋傳視技能啦。你能唔能夠喺個圖畫當中去揾到一啲眼見到嘅、觀察到嘅結構改變呢？而適應性特徵啦，都係嗰句啦，要將結構同功能拉上關係。跟住啦，就再問我哋有關於協作咯。今次就問啦，線粒體同埋粗糙內質網如何去協作？咁過往嘅題目有冇問過有啲有關於協作嘅題目呢？梗係有啦。過往係曾經問過特異性身體防衛同埋非特異性身體防衛之間嘅協作嘅，亦都有問過喺個葉綠體入面，喺個 Follicle 類囊體同埋個 Stroma 基質之間嘅一個協作嘅。而今年有一條有關於呼吸作用嘅 MC 呢，其實就係想考下細胞質同埋線粒體之間嘅互動咯。咁啊，過往都有唔少題目嘅，今日快手睇下片，溫下書啦。呢條題目你又覺得難定簡單呢？喺留言區留低你嘅睇法。Hello student, I'm Mr. Leung. So after we talk about the MC question, so let's talk about the long question. Paper 1B question 1 is about cell differentiation. Last year, the performance of the candidates is regarded as good. So let's see, is it this question quite easy for the student to work on and perform well? So the electron micrograph showed the appearance of a B lymphocyte before and after differentiation. So you can see the labeling of the organelles nucleus and after the process differentiation. So actually, what is these cells? It is a plasma cells. In the question, it's black and white picture, but in our textbook, we have the colored picture. So you can see that the highly folded structure, what are they? This small circle, what are they? So the question will ask you later. So let's talk about part A. State a stimuli which trigger this differentiation. Actually, for this question is checking out the concept about the specific immune response of the B lymphocyte. We can know that after the activation by an antigen of a pathogen, B cells and T cells respond differently to eliminate that type of pathogens. You can see that what stimulate, what activate the B lymphocyte? They are the antigens, pathogens, vaccines, or the bacterial or viral infection. Surely we know that antigen, that's the stimulus, but for the other answer, they are also correct. Because on the surface of the pathogens, maybe bacteria or the virus, there are antigens. And for the vaccine, though it may contain the weakened pathogen, but on the surface of the pathogens, there are also antigens. Therefore, them can stimulate the B cell and trigger the differentiation. And then for part B1, in this differentiation, apart from the nucleus, states two observable structural changes which are important to the function of the differentiated B lymphocyte. For this question is checking us as skills, you need to identify and describe the two observable changes. And then the question checks us the concept about the function of the B lymphocyte. But what is this B lymphocyte, right? It's the differentiated B lymphocyte. It is a plasma cells. Plasma cell release antibodies which are made up of proteins into the blood to act against pathogens that have not yet entered body cells. Therefore, for the concept checking, it also asks us to realize the adaptive features of the B lymphocyte after differentiation. So you can see that for this small circle, this pale brown circle, so they are the mitochondria. So after differentiation, there is an increase in the number of mitochondria. And also you can see this highly folded structure, they appear after differentiation. They are the rough endoplasmic reticulum. So we can also see the increase in the amount of the rough ER. So then for part B2, explain how the two structures mentioned in part 1 work together to bring about the function of the differentiated B lymphocyte. So for the concept checking, it checks us to do two things. First is about relate the functions of the two structures, and then match the function of the two structures for the differentiated B lymphocyte. First of all, we do the matching first. Mitochondria provide or release energy. That's the function. Rough endoplasmic reticulum to produce the protein, and protein can be used to make the antibodies, right? So we do the matching of the function first. And then we need to relate these two functions of the two structures together because they work together. So I need to talk about that. Mitochondria provide or release energy for what? Oh, for the production of antibodies at the rough endoplasmic reticulum. So you can see that if you present your answer one sentence by one sentence, mitochondria provide or release energy, full stop. And then the rough endoplasmic reticulum is responsible for the production of antibodies, full stop. You are not answering the question because you never talk about the two structures work together. Your presentation just means that they work separately. They do not work together. Therefore, you need a conjunction to connect these two functions together. It's just like that when we are doing the comparison question. So we need to say that A is da da da, but B is da da da. So you need to make it in one sentence and then put them together for the comparison. And then let's talk about the curriculum mapping. The question starts from the B lymphocyte, and then it checks us the concept about the differentiation and the organelles. For the differentiation, B lymphocyte could be differentiated into plasma cells to produce an antibody, and also for the memory cell to remember the antigen to provide us the immunological memory. And for the organelles, it checks us the synaptic skills. How can we identify and describe the observable changes? And then it also checks us the adaptive features of the organelles. We need to link the structure and the functions together. And the question also asks us about the collaboration of the organelles. Mitochondria, how can they work with the rough endoplasmic reticulum? In the past, there were questions asking the collaboration between the specific body defense and non-specific body defense. And for the 2016 question, is asking about the collaboration in the corpus, the stroma and the thylakoid. And in this year, there was one MC question asking the respiration. actually is checking us the concept of the collaboration between the cytoplasm and mitochondria. Watch the video and do the revision. And what do you think about this question? Is it easy? Leave your opinion in the comment section.